get feedback on the mic.
Oh, okay.
Is there mics?
I'm not.
It's going to be talking about the emerging CNS experimental therapeutics paradigm and the DCRI Neurosciences medicine program. Couple of notes before we get started. We are streaming this, we are recording this, so if you have comments or questions, please speak up. The mics in the ceiling will take care of that. Andy is currently Professor of Psychiatry and Behavioral Sciences here and Director of the Neurosciences Medicine in the DCRI. Also Director of the Sleep Research Laboratory, Insomnia Clinic, Quantitative EEG Lab, and Division of Brain Stimulation, Neurophysiology. I think of all of those as fitting closely together. Attended Massachusetts Institute of Technology for undergraduate and a master's degree in biomedical engineering. Then came here for a medical degree and psychiatry residency training as well as his fellowships. He's on several boards and journal editors. His primary research is related to pathophysiology and treatment of sleep disorders, mood and anxiety spectrum disorders. Please.
Thank you. This is my first time addressing this conference, and as Director of Neurosciences Medicine, I thought it was fitting to give some context for one of the major ways that I'm trying to develop the neurosciences medicine therapeutic area and the context for that. And this approach and this shift in the field is called the experimental therapeutics approach, or experimental therapeutics paradigm, a term coined by the National Institute of Mental Health Director and his team. And we will talk about what that means. These are my financial relationships. So I'm going to talk a little bit about what that paradigm is and what's driven it, which is a crisis in drug development, in CNS in particular, and what NIMH's solution to that is, and how that is an opportunity for us in DCRI. So it is striking, if you're in the field of related to neuroscience, how incredibly productive basic research has been. There's really been a tremendous explosion in many areas of basic research related to our field, and yet there's really little to show for it. We have developed very few important new therapeutics, and many of the companies that have traditionally developed drugs for a variety of problems have really had a drying up of their pipelines and a shift away from neurosciences. Let's see. And the drugs that we do have are not working so well in at least the psychiatric side of neurosciences. There was recently a large sort of practical trial done looking at how antidepressant drugs work in clinical practice, and only 40% of people responded to 2 adequate trials of antidepressants. And even after 4 trials of adequately administered, that is an appropriate dose for an appropriate duration of time, we only have 70% response rate. We have a lot of people who are failing to get better, and the development process is really dramatically going downhill. If you look at this from the long-term view historically, it is quite striking. Since the 1950s, in 2 of our most important areas in psychiatry at least, and I apologize that my view of the world, even though I represent neurology and psychiatry broadly, is oriented more towards the psychiatry because that's what I have worked in historically and know most about. But these concepts apply to both. The areas are depression and schizophrenia that I'm referring to here, where we really have only ever had one mechanism that has worked in either of those, inhibiting the reuptake of serotonin and norepinephrine for depression and blocking the D2 dopamine receptor for psychosis. So if you look at areas on a bar graph comparing what we've had in the 1950s to the present, looking at the depression and schizophrenia, it is dwarfed by stuff like heart disease, which DCRI has largely been responsible for actually, and has pushed the envelope to a great degree and brought tremendous therapeutics to help many people, whereas in psychiatry at least we are lagging behind, even though the science is moving forward at a rapid rate. So this is just an example of some of the innovation that we have seen historically. So it's a new antidepressant. Instead of swallowing it, you throw it at anyone who appears to be having a good time. That's about as good as we're doing. Literally, we are developing drugs that seem like they're good ideas, but they just never pan out. And it's not, the problem in innovation is not because people have not tried. There's been a huge investment in CNS, and yet there's just been little to show for it. And the costs just keep going up and up. I mean, this is a way outdated slide, but it's the only one I could find, looking at the amount of money that's been spent by the pharmaceutical company in drug development. It's going up every year. NIH funding is going up every year, and yet the number of new entities that are coming out is going down. Now, this is a very, very complicated slide, but the point that it illustrates, if you have great vision, is that in order to get one drug out the door for a company, it's estimated, and this is across all areas, this is not psychiatry or neurology or anything like that. You got to start in attempting to screen targets around 24.3 drugs, right? And once you get into clinical phase, you've got to take 8.6 forward to get one. And the cost, roughly, when this slide came out, which was 2010, was $873 million. And if you convert that into how much it costs, because generally people take loans to cover that kind of cash, it's like 1.778 billion. This is, very difficult to accept and to be able to function in a way, an environment of profit in that kind of context. So this has been a real crisis, and particularly people have had tremendous concerns about phase 2 and phase 3. That's the most expensive part of it, and we have 66% and 30% failure rates in those phases. Boy, you get into phase 3 and you have a failure, that's a huge hit, right? And this is overall, and in CNS and oncology, the rates are worse to a significant degree, and it's thought that's because you're trying new targets, and in psychiatry, I think that one problem we have is that our animal models are poorly predictive of our human diseases. Just to give you an example, one of the best models for major depression is you take a rat and you put it in water and you wait to see how long it swims before it gives up. That's what we do to test antidepressant drugs. And it happens to be predictive of the one mechanism that we've had for forever, which is selective serotonin reuptake inhibitors have efficacy in that model. But nothing else that has efficacy in that model seems to work in humans as an antidepressant. So we have an assay that is keeping us in the same place. Just to give you an example of what this kind of real-world examples of this kind of catastrophe. One example is a drug agomelatine, which is actually approved in Europe, interestingly, for depression. And I just have a few depression examples here because it's an area I happen to know well. But this is a drug that works largely via melatonin and serotonin type 2A receptor antagonism effects. And it has little side effects. It's helpful with sleep. It was approved in Europe. And Novartis acquired it to develop in the United States. And they did phase 3 trials, and it both failed and to show efficacy and had some concerning adverse effects related to liver function. So interestingly, they invested a huge amount in that, and it wasn't long after that Novartis just shut down their neuroscience unit and basically just decided to pull away from CNS drug development. And they are doing some now, but not very much, very little. Another example was a nicotinic channel modulator for depression, and AstraZeneca paid $1.24 billion for the rights to develop it, and they put it up front, $200 million, and it had great results in phase 2. So everybody was excited about it. And it failed 4 trials in phase 3. And AstraZeneca said, okay, that's it, we're out of here. I mean, you can't. How many hits of that magnitude can you sustain and continue to do work? Not a lot. And another example is a neurokinin 2 receptor antagonist, Sanofi-Aventis. Great positive phase 2 study, failed phase 3 clinical trials. Sanofi-Aventis is basically out of the space as well. And I think you can, the pattern here is that the phase 2 studies keep telling us that these drugs are promising, and the phase 3 trials, which is where you spend the huge money, is where things fail. That is intolerable. Just a couple examples in other areas. This is a very famous one. Lilly had a mGluR2 3 positive allosteric modulator that for schizophrenia, positive phase 2 studies failed phase 3, and for Alzheimer's, these are two areas where there's been a number of failed trials that have really been devastating from a point of view of finances for the companies. But Alzheimer's drug also, there's been several of these, failed to meet phase 3 endpoints, and what we're seeing is that pharmaceutical companies are really decreasing investment in neuroscience drug development, and for all the reasons that we've identified. So, to my count, AstraZeneca's cut back, Novartis has closed their facility for CNS, Sanofi Aventis is out, GSK is out. I mean, I don't know about people in this room, but I used to...
hired several of those people.
Yeah, right. So I used to work a lot with people at GSK, and I haven't worked with them in, 4 or 5 years now. They just have nothing going on in neuroscience. Zero. It's really striking. Pfizer, Merck have cut back. Though Pfizer and Merck and some companies are creeping back, and I'll tell you why I think that's happening. But everybody's also more risk averse. So what used to be the case is people would have a drug and they'd try it in one area in psychiatry, let's say depression, and if it didn't work, they might try it in schizophrenia, or they try it in schizophrenia. If it didn't work, they try it in depression. If it didn't work, they try it in sleep. And now, or substance abuse or something. And if it doesn't work in one area now, it's dead. They take one shot at it, and if it doesn't fly, it's dead, and then they let someone else out, they license it to somebody else. This is definitely, I've talked with the people at Lilly, and in fact, I'm working on a study with a drug that is very promising for mood and anxiety disorders in an NIH study because they weren't willing to take the risk once it failed substance abuse. They just said, we're shelving it. That's it. It's a great drug. The early phase work on it is really promising and exciting, so much so that the NIH said, we want this drug to be studied. Lilly wasn't willing to do it. They gave us a drug. They said, you can do it, but we're not going to do it. So there's a lot of lost potential here, and it's a crisis because the drugs we have, at least in psychiatry, don't work so well, and our patients need better options. So this is a dialogue that has not escaped the attention of regulators and the administration of the NIH. And the National Institute of Mental Health basically has proposed a dramatic emphasis on what they think is a solution to this problem. It's remarkable how strongly they have shifted their focus and invested in trying to fix this problem. And the focus of this is on solving what they see as drug development flaws, that is early phase methodology. The pattern, remember, we keep seeing that is killing companies, is that they have a promising phase 2 trial and they have a failed phase 3 trial. That you just cannot sustain that. So how do you address that problem? Well, I try to identify what the problems are in the methodology, and a number of them are contributing to the waste of money and the failures. One of them is that oftentimes people study drugs. Let's say that you're interested in something that blocks the serotonin type 2A receptor, but you don't bother to demonstrate that the dose you're studying actually robustly blocks that receptor. So they do an early phase trial, and on clinical data, they choose their dose, not on something like PET receptor occupancy or some kind of biomarker. And that's a serious problem. As we'll see, those clinical endpoints are very unreliable. And so what happens afterwards is they have a negative study, and then they say, well, if we'd only studied a higher dose, then they do another study at a higher dose. Or it was too high a dose, and they study a lower dose. It's really, that's wasting huge amounts of money. And different companies are wasting money because one does a trial, they fail with that drug in one mechanism. Another company does another study, same mechanism, similar kind of outcome, and none of them know if they're actually doing anything in the brain that they're intending to do. The other problem is that in psychiatry, at least, most of the studies that are done in early phases don't test a specific hypothesis. They want a drug to work. That's not a hypothesis. They say, if this drug improves something, or an endpoint, then we're going to call it effective. But that's really not a viable thing. You can have a positive outcome on a scale for many, many reasons, right? But the point is, if you say, I think this drug is blocking a certain receptor, then you really should test the hypothesis that you achieve the desired effect in the brain or behavior or something that is relevant, and not have a sort of a non-specific hypothesis that I want to see some benefit on a scale, which is something that I think we now appreciate is very unlikely to replicate, especially the way things are done, and I'll get back to that in a minute, in phase 2. So if you don't test a specific hypothesis, then you're not going to really establish proof of concept, that is, that your drug is achieving a desired effect in a region of a brain or some circuit in the brain or something related to the brain before you go on and try and test it in a clinical trial with a clinical endpoint, and failing to do that really sets you up for a problem. So phase 2A is the gate to this huge expense, and you need to know, is this likely to improve some FDA-approved endpoint? What are how are you going to design your trials, and is there commercial potential? And these phase 2I trials have substantial flaws. I've already alluded to some of them. But the biggest flaw in them, I think, is that they use these clinical endpoints, which we know are extremely variable and not capable of providing a reliable indication in studies of doable size. So if people want to do phase 2A studies, let's say they want to put in 100 people in it, 150 people in it, but when they get to phase 3, they use the same outcome, and they know they have to put in like 1,000 people in the trial to be able to have adequate power. Why would they think that when they did a previous study and they had 100 people, that was an adequately powered trial? It wasn't. What they're doing is a mini underpowered trial using clinical endpoints that give you an unreliable outcome. So let's say a depression scale. They'll do a trial and they'll do a dose response study with a Hamilton Depression Rating Scale, and they'll see some effect there, and with 90 people, and they'll say, okay, that's positive, we're going to go down, and they screen a bunch of drugs, and one of them has this positive dose response, and then they take that one forward, and it was just a chance finding, right? They didn't have anywhere near the kind of power that would have been needed to reliably detect an effect. And so they then fail in phase 3, where when they actually do have enough power to detect an effect, and we keep doing this over and over again. It leaves us vulnerable to bias. You do a 90-patient study, what's the chances that something affected the active condition versus the placebo in that trial with that drug that just led to a false positive outcome? Because people do 10 of those studies and they take one forward, right? And so your odds of getting stuff by chance start to go up and up and up. So you're vulnerable to bias, and then the other problem is that we're using, in psychiatry at least, diagnoses that have fundamental flaws. That is, it's not all clear that there is such a thing as major depression. This was diagnosis in psychiatry started because of the need to bill, largely. And there were a group of people like us sitting around a table trying to figure out what the diagnoses were, and we hone it every year. But if you look at it, there's very little science behind those diagnoses. And they're really a serious problem. That is, it's not at all clear that there is going to be a drug that targets some of these syndromes because they're heterogeneous conditions. Look at the diagnostic criteria for major depression. It asks you have to have 5 of 9 symptoms to qualify. So that means you can have 2 people and they can only share one symptom in common. Let's say it could be poor sleep, and they both could have major depression. One could be anxious and fidgety and very and having tremendous worries, and another one could be psychomotor retarded and feeling sluggish and really sad, and they are the same disorder and they look completely different. There's so many ways that people with depression can look different. So these are consensus-based, they're not science-based. They don't appear to predict treatment and response. We don't have good animal models. We don't have understanding of pathophysiology. There's no way we can develop drugs based on these things. It just fails. I think that there are many, many, many problems with this that have been identified, and this has been known for years and years. I was giving a talk related to this at an institution where the NIMH director from 20 years ago, 30 years ago, something that was in the audit, I think it was about 30 years ago, And he said when he was director, his primary initiative was to do away with this DSM thing and to come up with a dimensional system for diagnostic structure for research. Didn't happen, but it looks like it's happening now. So here's we came out relatively recently with a new version of the diagnostic manual, the Diagnostic and Statistical Manual 5, DSM-5. And here was what the field trial showed. So schizophrenia, one of our most important diagnoses, a kappa is only 0.46 on diagnosis, and the paper said that was good. So I think good is the new bad. Major depression, this is scary, 0.28. Okay, and at one site they had a 0.13. They said this was questionable. I mean, to me, this says this is a problem. This is a field that is in serious trouble, right? We can't agree on what a patient has even. We had generalized anxiety disorder, 0.20. I mean, this is one of the most commonly diagnosed things, generalized anxiety disorder, major depression. And they said this was questionable, and I think it's unlikely you're ever going to, that means nobody's agreeing at all. Right? And then we have mixed anxiety and depressive disorder. This had a negative kappa. And is that even possible? Can you have a negative kappa? I'm not sure. Maybe it was an error in addition or something. But, I mean, this is pathetic, right? I mean, and this was published, and the field has just gone on and kind of said, all right, this is what we're going to continue to do. I don't see how this could be useful for anything. So NIMH's solution involves addressing a number of these problems. One of them is to try and adopt a quick win, fast fail approach, which really means a fast fail approach. That is, given that the vast majority, I'm going to go back to the other things on the previous slide, but since the definition is here, I might as well go ahead for that first point. So since we're failing so many drugs and we're doing it late and inexpensively, can't we fail them sooner? Can we figure out a way to fail them with fewer patients and more effectively, and therefore save money and hopefully make it, drop the amount it takes to develop a drug from 1.8 billion to something less. And so that's one thing it's targeted to. The other is doing away with that DSM-based drug development and using a dimensional framework and employing biomarkers for those dimensions, because the biomarkers are assumed to be closer to the biology. So if you, let's say you're trying to block a serotonin receptor, and we know that there are serotonin receptors in a particular part of the brain, and your hypothesis is you should shut down activity in that part of the brain. Instead of saying, let's look to see if it changes the depression scale, you should audit. Shouldn't you audit first, see if it changes that part of the brain? And so that is something you're likely to be able to do with fewer subjects at lower cost. And if you don't do that, then you're done, right? That's their model. Smaller number of subjects, more rigorous way to fail a drug. So the way to do this, though, is you want to shift your money earlier. Identification of drug targets, and then do rigorous, excellent early phase studies, and then so that if you do go on to phase 3, you're doing it with some confidence and with a higher likelihood of success. And when you fail in phase 1 and phase 2, then you feed that back to help you learn about what you should do differently. And your studies in phase 2A should be designed to give you a capacity to make a rigorous go, no-go decision. That is, if this study shows that we cannot reject the null hypothesis, then we're done. We don't do another study at a higher dose or think we're going to do some other tweak. And the idea here is to test a specific hypothesis, use biomarkers to do it, and that's a challenge. Developing biomarkers for psychiatric illnesses is quite difficult. But one of the reasons this has failed is because we've had diagnoses that nobody agrees on. I mean, how can you possibly believe that depression research could have been done in any valid way given what you just saw? A kappa of 0.2? It's ridiculous, 0.28, something like that. So, so the dimensional framework actually provides an opportunity here that may allow us to do this in a better way, and I'll get to that in a minute. But this is just one person's conception of it, that is that you're the idea is to sort of oscillate in the early phase and only go here infrequently beyond into phase 3, where you do your proof of concept studies, you feed that back to your drug discovery process, and you keep doing that until you get something that is in a robust confirmation of proof of concept, and then you go on to phase 3. And it's estimated, so Steve Paul, who did a lot of these, I think he was CEO of Lilly for a number of years and has been an innovator in this field and wrote about this and along with the NIMH director, and is actually a consultant to our DCRI FastMOS project and has been very helpful in thinking about this. He wrote an article suggesting that there's an amount of savings here would be about half. So cutting from 1.78 billion to whatever it would be 800 million is a huge saving for a company. The diagnostic issue. So this is a dimensional classification system where this is one problem that's always bothered me about psychiatry. We've had these categories, and so you see a bunch of people who don't fit into, we can't talk about them. So if you don't meet one of those diagnostic categories, you don't exist in psychiatry. We don't study you, we can't say anything about you, we have no model of normality or even kind of normality. The idea here is that if you look at dimensions, everybody exists somewhere on that dimension. You can study everybody, you can study the path in and out of illnesses and things like that. And it just makes so much more sense, and it aligns much better with the available neuroscience data. And it turns out that we have lots of good animal models for many of the human things, like a simple thing is social things, like a social yawn. Monkeys, if one monkey yawns, another monkey yawns. Humans do the same thing. You can look at this as it's relevant to disorders like autism, right? That goes away in autism, and we can test circuits in the brain for monkeys that might, change that. I mean, just a simple example, but lots of the entities that we study now in this dimensional framework, this Research Domain Criteria Project, RDoC, are things where there are good animal models for it. That's how they were defined in the first place, so it was built in by design. RDoC was started by the NIMH. The director of the Adult Translational Science Division took this on as his project and working with the NIMH director and rolled it out. And now it is basically the requirement for NIMH grants to use this framework. They basically don't fund anything in DSM anymore, which is causing a huge shakeup for the field, and it may be pulling the cart a little bit ahead of the horse, but they want to pull the field kicking and screaming into this model because what we've had has just been useless. And I think the NIMH is very frustrated because they've done a lot of studies that have led to non-definitive conclusions because a lot of people have been using this framework that gets you nowhere. So the hope is that this will at least be a research structure and maybe eventually more than that. So the way this is defined, just to give you a little flavor of it is, and it's hard to use and it's clunky and it's not well flushed out yet, but the whole idea is that this is a living system that is being added to every time a new study is done, which is what it should be, right? I mean, if you have a static diagnostic system, you're dead because we don't know it all, right? In fact, we know very little, and that's what this shows us. So the idea is that you have a construct, which is what they call it, which is a dimension of behavior that is relevant for some understanding some aspect of human function or pathology, and it's sort of the fundamental unit of this system. And these things cross current diagnostic boundaries. For example, the study we're doing in this framework now is related to the capacity to experience pleasure. All right, that is impaired in people with schizophrenia, major depression, generalized anxiety disorder, lots of substance abuse people, post-traumatic stress disorder. I mean, so it cuts across those DSM categories, but it's a phenomenon that you see across many different kinds of situations and is relevant for study in all those situations because it looks like the underlying neurobiology is the same. It has to do with the ventral striatal reward circuitry and prefrontal cortical reward circuits and how they function, and there may be problems in these disorders in different parts of those systems, but you're studying the same systems. And so then there are animal models for this, right? You can lesion the nucleus accumbens in the animal and you get a loss of capacity to experience reward, or you give animals cocaine and you can ramp up their experience of pleasure. I mean, there's just lots and lots of ways to study these in valid models, which we just never had. And the idea is that we want to integrate the genes, the cells, circuits, all the self-reports and clinical outcomes for these dimensions, and sort of flesh out a full system, which is a long way off. They identified 5 domains, what they call, and they needed a marketing person to do this. They really did a terrible job in the naming and the selling of it. It's awful. Like negative valence systems, positive valence systems, cognitive systems, systems for social processes, and arousal regulatory systems. It's unfortunately, it looks dense and difficult to understand, but once you get into it, you can work your way around it. So if you just look at reward, the ability to experience pleasure, that whole domain, this was done as a consensus document also. So, unfortunately, we have limited capacity to develop things without using that kind of process. But it was done with the intent of bringing together researchers who had done research that was relevant to defining the key dimensions. And so RDoC's constructs are basically a row in a table, and there are multiple dimensions to the reward process. One of them is the capacity to value rewards. That is, can you tell whether something's going to be rewarding or not, right? Or like, are you about to get a punch in the nose or nothing, or chocolate ice cream, and should you do one of those, or approach one of those or the other, or and so that's your ability to tell whether it's there. And another dimension is how much you're willing to do in order to get a reward, and then your ability to expectancy and action, decision making based on reward, learning based on reward. So there are all these pieces to it, and in our study we're studying each of them. But you can tell what we're trying to do, or the field is trying to do, is to map out all the key biology, genes, molecules, cells, circuits, physiology, self-reports, and paradigms for studying. And you notice the genes are empty. Hopefully we'll get there. A lot of it's empty. But there are some where there are relevant genes. These are other aspects of reward as well. I didn't mean to have you try and understand any of these specifics, just to give you a flavor for what this is like. But NIMH has really said, this is what we're going to do. They have said, the NIMH director announced, I was there when he did it, and it is borne out, that they are not funding any research that doesn't employ this new paradigm and doesn't employ the RDoC. It was a huge shock to people. It just was like out of the blue. And the model, this experimental therapeutics model, is that you have a biomarker-based development approach where you test a specific hypothesis related to engaging a target by using some intervention that at that dose robustly engages the target, and then you test the proof of concept in a hopefully smaller study than we used to do in phase 2A or comparable, but get a definitive no-go or go decision from it. So I think that I covered this. Okay. So NIMH has, one of the ways we know about this, because it's hard to sort of get your hands around what they had in mind, is by their having rolled out a series of RFAs related to this model. And we were fortunate to get one of them. They did 3 sort of flagship programs, one in mood and anxiety, one in psychosis, and one in autism. And we here at DCRI have the mood and anxiety one, which has been tremendously valuable for learning about what they had in mind. And the remarkable thing is, when this started, which was about 3 years ago, they didn't quite know what they had in mind. This has actually been an evolving process, and they've been sort of coaching us and helping us figure out how to do this. And they said, okay, we want you to tell us what projects can be done in this model, and then we're going to use that to help us figure out how to move forward. So we, after like a year and a half of struggle, and it was intense, hard work, we came up with a project that they eventually were happy with, and then they rolled out a series of RFAs in that model. They said, here's what we want. And that gave us a big advantage. We were able to get another grant because we knew what they were looking for and had experience with this model, which is what I'm going to get to about how this is an opportunity for DCRI. We have a 6-site network that is doing work in this area, including a bunch of people with fMRI, PET, EEG, a lot, This is largely DCRI people on the administrative team, where the goal is to choose a target that is promising in some way because of preclinical data. You have a molecule that you know engages a target, you establish a dose that robustly engages it, you use an RDoC construct that's relevant, like the capacity to experience reward or predict reward or something like that. You identify outcome measures and you design your trial to give you a definitive fast fail model, a go, no-go solution at the end, or go, no-go decision at the end of it that's rigorous. And we're using a Lilly drug in a trial that's going on now that I said before they had dropped, but it's very promising. We did PET to establish a dose that we know robustly blocks. It's a kappa opiate receptor antagonist. We know we block the kappa opiate receptor at 90% occupancy at trough. I mean, we're nailing it hard. And then we're testing a fMRI-based hypothesis on affecting a specific circuit. And we're hoping that we will be able to definitively say, yes, we affected that circuit, or no, we didn't. And if we didn't, then that drug will be dropped for that purpose. And reward is what we're looking at. Now, someone may say, okay, we can still develop it for depression. Obviously, that would be vulnerable to all the flaws that have existed in the past. A reward is not depression. Depression, in my view, doesn't exist in the world. But the capacity to experience reward is something you can measure in anybody. And there are lots of paradigms for doing this. You can measure it in monkeys. You can measure it in rats. You can measure it, I don't know about insects, but you can measure it in a lot of things. So we're using a task-related fMRI as our primary outcome. And that's a shift in the model. We're not using a clinical outcome. They're in the study, but they don't count. And the problem with this approach is that we don't have biomarkers for a lot of the things we'd like to have. We don't have molecules to target a lot of the targets we'd like to study. And so there's a huge pressure to develop this stuff. And that's what a lot of the NIMH RFAs are on now, coming up with methods for doing these kinds of studies. There is another risk of failure in this effort, and that is that you could have false rejection of a good drug, but I think the risk of that is a lot lower than the risk of accepting a drug falsely in terms of probabilities, but then also of cost to companies. In terms of the rate at which it's going to happen. But then there's also the problem that just because you move something to phase 3 because it achieved your hypothesis, you had verified your hypothesis, doesn't mean the drug is going to sail through the FDA and make it to pharmacies. And the problem is when you get to bigger studies, you may identify adverse effects that you wouldn't have seen early on and other sorts of things. To summarize, we've seen a rather dramatic shift of industry away from neuroscience because of the risks and the losses. And from my point of view, this is really a rare opportunity, a great opportunity for us, because people are now changing how they're doing trials. The NIMH is forcing us to do these trials in a different way. And as I say, all the trials are going to be carried out in this framework that NIMH supports. There's no question about that until the NIMH director leaves, and of course that may all change. And industry is increasingly moving towards it, although industry is moving towards it very carefully and slowly because FDA endpoints are uncertain. We don't know, the path through the FDA is not established for any of these new RDoC endpoints. Though there's an FDA consultant on my team who, and NIMH has been working with FDA, and they're very happy to look at endpoints related to this RDoC, this new RDoC model, and they've been very encouraging of it. There was just a consensus conference on looking at cognition as an outcome that could be a target, an approvable target in schizophrenia and in major depression. Well, it would cease to be in a diagnosis, but it would be across diagnoses. So the field is moving there. And I think that there's the shift in this field is including biomarkers and high technology, technology-intensive methods in early phase work. And really, that's where we have an opportunity, I think. That is, we have the opportunity to develop these methods. There's a huge amount of work to be done to figure out how we are able to establish target engagement and figure out how to do these trials in a way that gives us robust answers. And really, in psychiatry anyway, the clinical trials have been done by for-profit CROs. I'm sure you're familiar with these companies and how they work. But these companies are not in a position to be able to function in this new space, right? They don't have experience with it, they don't have the expertise, and to do a trial here, you're basically going to have some biomarker capacity or the capacity often to develop or plan a new biomarker program that will be needed to help a company move forward with a drug. And so that means an opportunity for academia, and particularly I think for DCRI, which is that rare mix of academia and capacity to manage studies. And I think we have that rare technical capacity, and I think that our experience already and the strengths of the faculty here and the resources we have really should put us at the center of development of CNS therapeutics. And I've already seen it happening, but I think the explosion is just about on the horizon, and hopefully will become the go-to place for CNS drug development in the future. That's all I wanted to say, and see if you have any questions.
Questions? And if you could go one more so that the people who are doing the text messaging.
Oh, there you
Yes. Please.
So this model, Andy, that you're talking about could actually apply to other therapeutic areas as well.
go. Absolutely. Well, so this was some of this was designed specifically to address problems in the CNS space, development space, but these are methods. Some of this is already going on in other areas. It's been going on. It's just we haven't been able to do it because we haven't had the measures and the technology capability, and and our diagnostic system has been dragging, it's been an anchor around our ankles for so long that we haven't been able to move forward with stuff like that. But for example, if you are in GI and you're studying liver, and you can get a liver biopsy and establish that you have what your pathology is, and then you can do another one and show that you've affected it. You're already doing what we're trying to do. Though I had a talk about this with Rob Califf some months ago, and he has been having dialogues with the NIMH director about this idea, and he said, their biomarkers aren't perfect, and we have biomarkers, and we still have problems with developing studies, but I want to have those problems, right? I want to have the problems that GI and cardiology is having, and not that my diagnoses are baloney, right? Not that we don't have a way to measure outcome that's valid, or we can't figure out if we're affecting the part of the body we're hoping to affect. We get past those hurdles. I'm happy to deal with the limitations of biomarkers that are inevitable.
Yes.
So it looks like a majority of these drugs, especially for a large pharma, are failing from phase 2 to phase 3, right?
Right.
Okay, well, as you guys know, when you go from phase 2 to phase 3, usually you're adding another geographic region, right? So you'd have US, Europe, and then India or Singapore, China.
Right.
So has there been any analytics done, backwards looking or anything like that to show the bias that is actually created in those third regions?
Well, so there's a fair amount of interest and work going on looking at site behavior and how that affects outcomes, because in CNS studies, at least in the ones I'm familiar with in psychiatry side, we'll have like a 400 site study, and a lot of the work that comes out of many of those sites is not good quality by metrics that have now become available, and they're starting to look at these, and now they're rating sites and dumping them. But historically, these sites have been putting in people who don't have diagnoses that are appropriate, that are not meeting entry criteria, and a lot of problems. And and there is data supporting that that affects outcomes. And so increasingly there's a move in that direction. But I had one very vivid example of this when I was on an advisory board for a company developing a drug for psychotic depression, I think it was. They started out with a consortium of, I think, 7 or 8 academic sites. And they did their phase 2, and they had a very positive phase 2 outcome. And psychotic depression, unlike major depression, is actually a much more valid diagnosis. It's a very specific thing, and there are criteria for it that you can reproduce. I mean, it's much, much different. Psychosis is much easier to have agreement on when it's present than depression. So they then went into phase 3. They took the same sites forward, but their investors were not happy with the speed of enrollment, and of course they were trying to use the same number of sites and do a much larger trial. So they kept pushing and pushing and pushing, and finally the people who were running the study for the company said, okay, we're going to go and add sites, and they went to, I think, Eastern Europe and maybe India, I'm not sure where, but outside the United States. Finished the trial, and then they analyzed the data, and had they stopped before they added the other sites, they would have had a significant effect. The other sites added had like a placebo response rate of 70%, and the initial sites had a placebo response rate of like 25%. That's one example, but there are others, and I think that this is a huge problem in the field, and the biomarker-based research helps you with that because you do smaller studies, but eventually you got to go to a phase 3 trial with an NIMH, FDA endpoint, which may be a clinical endpoint, and you're going to need large numbers to address the variability in those measures. That's still an unsolved problem. But I think there's 2 things that people are doing. I'm sorry, Sarah, get your question in a second. One is they have metrics for site quality, and they're using that to choose sites. And the other is they're having central outcome ratings. So the sites don't rate outcomes. The patients get video interviewed by a central person, or the site rater gets videoed and that gets verified and things like that. They're doing as much as they can to verify that people meet entry criteria and assess outcome that way. Sorry.
It strikes me that a lot of what you've just described about sort of the historical and up to present time challenges with NSM research has been sort of an uncoordinated or a not very well organized approach, starting from not having solid science-based ways to diagnose patients in the first place, all the way up to how patients might be enrolled in a phase 3 trial at this point. How much, if any, impact do you think sort of stigma has with the diseases that you're studying on that, and is that changing?
Well, that's a really good question. I think one of the things that stigma does is it prevents people from getting treatment, so we don't necessarily have a representation of the full sample in our studies. I think that in the depression area, the biggest effect on stigma that I have seen was the kind of household name emergence or change, that Prozac became a household name, and people talked about Prozac, and people started talking about having depression, and it seemed to be okay to have depression at that point, and and that really changed things. A lot of people started to seek treatment. And I think we then had a much more representative population, both getting treatment and being in studies. But before that, the studies were done in hospitalized people almost entirely. They were more severe. The variability was decreased because the conditions were fixed, and the effect sizes were dramatically larger, actually, than have been the case since. Yeah, Peggy.
So what's being done, if anything? It doesn't sound like anything's being done. There was a movement a while back, years ago when you and I were working together, with Pfizer to try to establish objective anchors to a rating like the CGI. Has anything happened with that, or has it even been attempted?
Not that I'm aware of. There are efforts to try and come up with ways of rating outcomes for these RDoC dimensions that are anchored, but for a lot of this stuff, it's just not going to be possible because we don't have biomarkers for depression. I don't think we ever will. I mean, there are people who argue that we do, but I'm not seeing it. Yeah, please.
I don't know a lot about mental illness, but it seems like there's a whole spectrum of treatment that is outside of just a chemical.
Yeah.
And I'm wondering, is it naive to assume that there's a silver bullet that can address this? And how do you, I don't know, like when you think of cancer, you've got, surgery, radiation, chemicals. What kind of things can you bring to the table for the patient?
I apologize, I gave an overly narrow view of the world.
Mm-hmm.
This model is also being employed with psychotherapies and with non-medication interventions, like we're doing studies with games that are, affect brain function, right? And are being used to treat ADD or autism, whatever, and improve capacity to do things. There is a difference. There are some steps of the process that you can't do, but if you have a hypothesis that doing this game should enhance your working memory capacity, that's a really testable hypothesis, and that works just as well. But there is a huge development in non-medication therapies in this field, and treatment is becoming, and has been for a while, broad in that sense. Not quite in that model that you have these 4 components or whatever to your therapy. But I'm sorry I underrepresented and focused on the drug story because that's what's received a lot of the attention in the writing. But NIMH is not funding any studies with psychotherapies that don't use the same model either. It's the same story. And I apologize I didn't. Galen.
Andy, you made the comment that the absence of biomarkers and you didn't think that it would occur. And 2 aspects of the question. One is, what's your definition of biomarkers? And 2, why is it you don't think there's ever going to be any?
Well, so what I meant by that is that I believe that there is and will be biomarkers for the dimensions of behavior that are being defined that are now the focus of NIMH. But you missed a part of the talk where I went through the flaws with the DSM system. That the reason I don't think there's going to be a biomarker for major depression is because I don't think major depression exists, because it's a heterogeneous condition that 2 psychiatrists can't agree on for, hardly at all. You might as well flip a coin, right? So I think the problem there is you get a group of those people together in a trial and you look for a biomarker, and there's not an underlying unifying problem there. You've got some people who got one problem, some people have predominantly a problem with sleep, and some people probably have a problem with pleasure, and some more with anxiety, and you mix them all together and you look for one marker of a biological change that correlates with a symptom. That's what I mean by a biomarker. You're not going to find it because they don't all have one symptom. I mean, it's a mess. Yeah.
It almost seems like major depressive disorders is about 6 or 8 diseases lumped together, the way some people think about cancer being 200 different diseases lumped together.
So the NIMH's model, there is no depression. You can't find it. There are 9 dimensions or 15 dimensions that get pieces of it. And it is true. So when I've talked about this, but I'm not selling this story. It's the NIMH who are trying to sell this new dimensional model. There's a lot of pushback because people say, look, I'm in clinic every day and I see these people with depression. And the reality is I've thought about it because I've come through the same path of having been in clinic with people many years and used this model. When you can step outside that, you appreciate part of the reason you're seeing it that way is because it was set up that way. You're looking for these 5 out of 9, and if you looked at it in a different way, and I've tried to do that, I find that most people with depression have a predominant problem. Like, my problem is they don't experience pleasure, or they have predominantly an anxiety problem, or they have and there are other consequence symptoms that you can identify. So I think partly we've biased ourselves by creating a worldview that we then, we then try and employ, and in employing it, we sort of have convinced ourselves that that's what's there, but it's really not when you carefully look at it. I think it's the case is that every one of the people we see with depression has got a different mix of things, and that's also true with schizophrenia, and that's also true. And some people have cognitive impairment with schizophrenia, and some people don't, and some people, some people have problems with reward, and some people don't. So there are a mix of things, and that I think we're going to have to go through a dramatic change in viewpoint and perspective to be able to work in this model. But I think it's more valid. Yeah.
I understand the reasons for confusion, but one of the reasons for confusion should not be because we confuse symptoms with diseases.
Right.
I can certainly understand. I mean, to me, depression is a disease, and all these other things are symptoms, like you can't sleep, or you're unhappy, or this and so forth. So I can understand there's not going to be biomarkers for the symptoms, but to the extent, but surely depression there must be a disease that is depression.
Why do you think it's a disease?
Because I think that it's a clinical problem that people have because of some underlying pathophysiology in their nervous system.
But despite like 50 years of this, we haven't been able to identify the pathophysiology. And I think that part of the problem is that if you actually look at one slide I showed before, is that we can't, when you look at a person in front of you and you try and use the criteria we have to identify something, it turns out that our agreement on that is horrible. That is, we actually are not getting a consistent entity in our net when we use that diagnostic criterion. So there may be an entity there may be a disease there, but it's not as it's currently defined.
Well, I think to me, if you're talking about disease, the question is where is it? And my answer is it's somewhere in the brain.
Right.
So one way, if you take the analogy with heart things, that you then do these diagnostic tests with the heart. You look with some way. You look with an echocardiogram. You look with a magnet. Right. So if you look at the brain with all the various neuroradiologic or whatever techniques there are being able to image the brain, has anybody come up with some actual diagnosis, some abnormality in any of those ways of imaging the brain in people that have, if you just take people that definitely have depression, no doubt about it, another bunch of people who don't have anything at all that even suggests depression, and you image their brains by whatever radiologic or method there might be. I use radiologic in broad. With all the new things that are happening in neuroradiology, it would seem to me that there's got to be something in these people that have severe depression. There's got to be something in their brain that ain't terrific.
Right.
It's different than what's in the brain of somebody who has absolutely nothing to suggest depression.
So the problem there is that it's not really possible to get a group of people that everybody agrees on has depression. I guess you could take a very, very small subset of the depressed people and identify, and get very high agreement on it. But they're going to look different from each other, and to date there has never been evidence of a finding that replicated. There's 50 papers of findings of neuroradiology, but they're not replicating, and they're not replicating because I think the diagnosis is not a valid diagnosis.
Well, I think the
we've reached the end of our hour, so I'm going to take the moderator's prerogative and thank Andy for a great talk, very stimulating. Thank you very much.